And let me confirm everyone can see the screen, correct?
Yes.
Okay, let me go back to, okay. So I'd like to thank everyone for being here for the third educational seminar of our organization. We are now known as Just Humans, AKA formerly Advocacy Q+. Today we're here to talk about sex past the binary and establish a case for non-binary sex development outcomes. This is a research review paper that has been written by Just Humans, and we are seeking to publish currently in the Journal of Sex Research. Um, we have used this to test the hypothesis that sex characteristics are plastic traits. They're not binary, they don't have fixed outcomes, and to establish through that hypothesis test through research review that a new definition for the trans community as neuronally hormonally incongruent due to the loss of language here recently with how this community can be discussed. We're seeking to establish a more scientifically valid definition to maintain future ongoing research. So I'm your speaker today. I'm Rebecca Bishop. I have a master's in genetics from Clemson University Center for Human Genetics, a bachelor's in genetics from Clemson University, and I currently have 2 publications and 89 total citations. I'm currently working on 2 more publications, one in a previous lab I worked in, and the paper we're here to discuss today. I'm also the founder, president, and lead researcher for Just Humans, aka Advocacy Q+. So our seminar objectives for today are, as I've kind of mentioned, to look into the hypothesis that sex is a plastic trait and to establish a new definition known as neuronally hormonally incongruent. To do this, we're going to do 3 key things. We're going to understand the biological evidence that challenges the binary definition of sex. We're going to look at the role of genetics, endocrine system, neurobiology, and even the environment in sex differentiation. And we're going to talk about implications for scientific research, medicine, and policy from the findings of this comprehensive review. It's best to start with, how did a binary model of sex even come to be in the first place? This began in the 4th century BCE with Aristotle, who claimed that sex was determined by the heat of the semen and that males were the default and females were viewed as an incomplete occurrence from not enough heat being in the semen. This grew in Hippocratic and Galenic theories of male and female variations that all stemmed from a singular one sex model. And then throughout the 17th and 19th century, external anatomical observations began creating the idea that there was a fixed sexual dimorphism and therefore a binary. In particular, in the 18th century, Carl Linnaeus created the biological classification system that established this initial strict binary categories. Early scientists sadly built on this for a long time, and they continued to categorize sex using just external genitalia and reproduction alone. Eventually, we began moving from observations into genetics. In 1905, sex chromosomes were first discovered by Natalie Stevens and Edmund Wilson, who first identified the XXXY chromosomal systems. This further reinforced the strict binary classification of sex, sadly, at first, though it was a great turning point because this would lead to the loosening of that definition in time. These studies continue to exclude intersex individuals as well, counting them as anomalies despite their documented existence from a historical perspective. In the mid 20th century, we began seeing advances in endocrinology and genetics. In particular, Alfred Jost in the 1950s demonstrated that testosterone exposure to a fetus masculinizes the development of that fetus. As well, this sadly only further enforced the strict binary, but it began the movement of understanding that hormones in the endocrine system play roles in sex determination outside of just chromosomal and sex chromosome components. And in time, this would lead us into the understanding of the true variability of sex characteristics as they present across humans and the animal kingdom as a whole. We eventually learned how to start staining and actually imaging chromosomes from humans and other species. You can see here on the right what is known as a karyotype. This is a way of actually visualizing the diploid set of chromosomes inside of an individual's cells. You can see the 22 autosomes indicated and the 2 X chromosomes for this individual on the right. This finally allowed for determining that sex chromosome abnormalities were actively occurring within the human species, whether this was through different sizes in the X or Y chromosomes than should be seen, different volume within numbers of these chromosomes, absences of these chromosomes, and we'll touch into these different aspects as we move along. This slowly led to a shift in the emergence to understanding intersex conditions. And this occurred due to non-binary inheritance of sex chromosomes that could finally be seen visually. In the late 20th and early 21st century, we began to develop a better understanding of genes and their roles in sex determination. And a lot of these genes exist outside of the 2 sex chromosomes. So they're all what are known as autosomal genes. They come from those other 22 chromosomes. The SRY gene is on the Y chromosome and is known as the sex determining region of the Y chromosome. But we now know many other genes like DAX1, FOXL2, WNT4, and many others play crucial roles in sex characteristic and brain dimorphism development patterns throughout multiple stages in human development processes. Endocrinology and neurobiology built this understanding that hormonal influences on the brain created complex non-binary interactions and outcomes. In the 1990s and 2000s, we finally began seeing intersex visibility and medical reclassification start to take place as a whole. Medical professionals began shifting from saying things like disorders of sex development to variations of sex characteristics to start to take into account this complex genetic interaction of determination and the broader range of variability that we were seeing. This increased recognition that sex is a spectrum, that it's influenced by genetics, hormones, the environment, and complex neurobiological realities of natural human development. So what is the current consensus of science now? Science currently confirms that the binary model is a very simplified framework, and it does not reflect biological reality in its whole. Sex differentiation is now known to be a highly multifactorial, developmentally plastic process, and it's not as rigid as once believed. So to summarize all of this before we really move on, early scientists used external observations and excluded incongruencies to establish a binary perspective. Early chromosomal evidence reinforced the binary initially, and then later chromosomal evidence has revealed many non-binary outcomes. Modern genetics, endocrinology, and neurobiology have determined a simple binary framework to just be completely inadequate. This creates the idea that genetic and endocrine systems contribute to sex plasticity. We know sex differentiation is regulated by genetics, hormones, and the environment, that XY and XX binary models are incomplete without the reality of genetic variation in gene expression or endocrine influences on the developmental outcomes of those genetic variations. We're now going to move into how the SRY gene and other sex determining genes and hormones shape this developmental pathway, or pathways, I should say, not singular. The role of the SRY in gonadal differentiation is an important place to start. The SRY is known as the sex determining region of the Y chromosome. And it's a series of genes that are all highly linked together through linkage disequilibrium. And they work to activate a lot of other different genes through transcriptional activation pathways. One of the most essential genes activated in XY individuals that have an SRY gene region is the activation of this SOX9 gene that you can see here to the right. This little green circular arrow you can see under SOX9 indicates that when this gene is active, it creates a feedback loop with itself. So it further activates and maintains its own activity, and this leads to driving testicular development. As well, from this figure on the right, FGF9 and PGDS are autosomal genes that also further work in unison with the SRY genes to activate SOX9 so it can perform that cyclical relationship. For XX gonadal development, the SRY is not typically present, and therefore the other factors like FGF9, PGDS, and all these other genes also don't come in to help activate SOX9. These red lines with bars that you can see leading to SOX9 indicate that Wnt4 beta-catenin and FOXL2 are acting as what are known as inhibitors to stop SOX9 expression. So without the presence of these other genes to the left, these genes can come in and stop SOX9 from doing what it does and driving testicular development. And this contributes to driving ovarian development, which also further drives more FOXL2 activation, which further feeds back to inhibiting SOX9. Mutations in SRY or downstream genes can lead to 46XY sex reversal, where a female phenotype, so female genitalia, are observed despite an XY karyotype. So that staining image that we were seeing of the chromosomes would show an X and a Y chromosome, and yet we're still seeing ovarian and vaginal development. On the other hand, SRY is not the only determinant, as we've been discussing. Some individuals that are XY but do not have an SRY region still end up developing penile-based genitalia, which further reinforces that there are multiple genes playing effects here in how this development of sex characteristics occurs. So to look beyond the SRY at some of these other genes we've mentioned, DAX1, Wnt4, FOXL2, they all contribute to ovarian development, even in the presence of XY chromosomes. DMRT1 and SOX9 in particular support male differentiation, but loss of function of these genes, even in the presence of SRY, still leads to ovarian development. So, as well, chromosomal variations outside of that XX-XY binary outcome lead to disrupted pathways. There are two very critical ways of testosterone exposure to start looking past the genetics of this and into the hormonal pathway of how sex characteristic development occurs. The first surge is during the fetal stage, and this can result in masculinization of the brain and early reproductive structures. This produces a sexually dimorphic masculinization of the brain that presents as a mosaic in all individuals. And a mosaic means that all individuals have varying different cellular levels of masculinization and feminization to specific regions of their brain. And we're going to kind of look into that in detail as we move in. What does that mosaicism look like? It's important to keep in mind that this isn't just within intersex and incongruent individuals, this occurs across the board. A second surge of testosterone occurs during puberty, and this drives secondary sexual characteristic developments. Disruptions in early testosterone exposure can lead to neurological feminization in an XY individual, further showing that this just is not a binary process. So we've looked at testosterone in some detail. Let's kind of talk about estrogen some as well, and its role in gonadal and brain development. In particular, testosterone is the starter of all these pathways. Testosterone activates a enzyme known as aromatase, and this is encoded by a gene known as CYP19A1. And this is responsible for converting testosterone into estrogen, otherwise known as estradiol. This estrogen can interact with two different types of estrogen receptors, alpha or beta, to cause what is known as transcriptional modulation. This is a fancy way of saying it changes levels of different genes being activated or suppressed, or different levels of expression even of those genes that we've been looking at that further modulate sex characteristic development. Estrogen is critical for ovarian development, and it influences sexually dimorphic brain structures. Overexpression of aromatase in XY individuals can lead to feminization. Disruptions in hormonal pathways in neuronally incongruent phenotypes, as well as intersex individuals, have been well documented. Androgen insensitivity syndrome, AIS, is where XY individuals develop female phenotypes due to non-functional androgen receptors. Congenital adrenal hyperplasia, CAH, is another one of these examples where excess androgens in XX individuals still contributes to masculinization within gonadal and brain dimorphism development. Finally, endocrine disruptors, which are human influences to the environment through chemical runoffs and productions and materials that we consume, such as BPA and phthalates, have known abilities to alter sex differentiation at this point. So a quick summary of all of this is that genetic and hormonal influences demonstrate that sex is not strictly binary. Understanding variability in sex differentiation has medical, psychological, and policy implications. So let's start looking beyond genetics and start really considering the brain and the environment and how it affects sex differentiation, both from a gonadal and a neurological perspective. Sex differentiation, as we've kind of been seeing and we're about to expand on, extends beyond chromosomes and hormones. Neuronal architecture and environmental factors play crucial roles in these developmental pathways. We're going to really quickly overview how these neurobiology and external conditions shape sex traits. Brain structures such as the hypothalamus, amygdala, and corpus callosum do show sex-specific, sexually dimorphic variations. So there's ranges in how masculinization and feminization of these brain regions occurs in all individuals. Prenatal testosterone exposure plays a crucial role in masculinizing the brain, while estrogen influences synaptic plasticity, which is the ability for neurons to change how they respond to one another and form new networks. Studies show that XX individuals who are exposed to high testosterone levels can develop male typical neuronal patterns, while XY individuals with androgen insensitivity can develop female typical neural architecture. And neural architecture is just a fancy word for saying how are all these neurons arranged, especially in between other non-neuronal cells like glia and astrocytes. And how do these neurons connect to each other and send signals back and forth? This challenges ultimately the assumption that genetic sex directly determines brain sex. Endocrine disrupting chemicals, as we keep kind of mentioning, such as BPA and phthalates, can alter hormone signaling. And we've looked at how hormone signaling drives gonadal and neurological development a little. So you can kind of go ahead and start, thinking through, okay, disrupting these pathways with external chemicals is definitely going to change the outcome of those pathways. And this leads to feminization of male offspring or other intersex variations. Prenatal stress exposure as well greatly affects cortisol levels, and this is being thought to potentially disrupt normal sex differentiation in the brain, and even in certain instances of gonadal development. To look past humans even and further expand that this is not unique to us as a species. These differences in sex outcomes, and in particular, this exacerbation of environment and endocrine disrupting chemicals, it's good to look at species like reptiles who have temperature dependent sex determination systems where higher incubation temperatures often result in female bias sex ratios. And even in normal situations, this factor alone shows that sex differentiation is not a binary, simple, singular construct, especially across the animal kingdom. And it highlights the role of environmental inputs in shaping biological sex beyond genetic determinants. Neuroimaging studies have revealed structural differences in regions like the amygdala and hypothalamus, as we've said. And we've already kind of looked at as well how testosterone and estrogen both uniquely shape brain connectivity and function. Neuroimaging studies have further confirmed this difference in shape and interaction through those neuroimaging studies. And this has resulted in us understanding that sexually dimorphic nuclei, which the nucleus is the center inside the cell that contains all the genes, and it's the little workhorse that has the blueprint for absolutely everything. But we've been able to determine that in the brain, there are sexually dimorphic nuclei sizes, and these occur due to different types of hormone exposures and levels. We kind of already discussed as well that prenatal testosterone exposure masculinizes the brain, and this influences both behavior and cognition. I want to keep kind of repeating and building on some of these things as we go. So I do apologize for some of the repetition at times, but I think it's essential and really keeping all the pieces of this story completely synthesized together as we go. We've looked at how estrogen affects synaptic plasticity, which once again was how those neurons can change how they link to each other and therefore how they pass signals along to each other. This is a process that's related to learning because learning alters synaptic plasticity and changes the way neurons influence each other. We also know that estrogen affects neurogenesis, which neurogenesis is the actual process of creating new neurons to replace old neurons. And we've looked at cases of hormonally incongruent neuronal development that challenges binary models. And we're going to dive into these further. Genetics and environmental variations influence sexual differentiation. Examples include climate and climate change, endocrine disruptors, and prenatal stress, as we've seen. Temperature-dependent sex determination systems in reptiles also confirms disruptions are occurring and that non-binary pathways take place naturally within determining sex characteristic development. Past this, even in certain fish species, um, availability of mates can cue natural life cycle switches known as sequential hermaphroditism. And examples of this are in clownfish and parrotfish, as well as a lot of other coral species who naturally transition from one sex characteristic development to the other as a natural part of their life cycle, as well as in response to environmental cues, showing that this is just a deeply multifactorial and complicated process. As we keep kind of saying, climate influences sex in reptiles and other species. You can see on the right the changes in different sex ratios relative to different incubation temperatures. Temperature sex dependency is common in reptiles such as turtles, crocodiles, alligators, and other cute little reptiles. And it often produces more females. This is further exacerbated by the fact that aromatase activity increases with temperature and boosts estrogen levels. This also leads to sexual dimorphic changes in human brains as well. As we've seen, aromatase stimulates estradiol, stimulates estrogen receptor binding, pathway goes on, yes? So aromatase being increased in individuals is going to increase the amount of estrogen in their brain and change the way sexually dimorphic brain alterations occur. Climate change may be skewing these sex ratios in vulnerable species. We looked at how endocrine disrupting chemicals interfere with hormonal signalings and linked this to altered gonadal development, reduced fertility, and feminization of male offsprings, which is something we're seeing in humans as well as all across the animal kingdom. We will, as we touch on to certain intersex individuals, this idea of fertility, especially as one of these altered consequences, will be really important in looking at how sex development and differentiation is so severely complicated. Studies have also been showing recently that declining sperm counts are and increased intersex traits are occurring throughout wildlife in the animal kingdom currently, showing that there's a large global response to what's happening right now and a plasticity in sex development to change in response to these cues. If sex characteristic development was binary, this would not be the case. There would be no modulation to outcome relative to these different factors. So let's start expanding the sex spectrum. Brain structure, hormone exposure, and environmental conditions shape sex traits. Scientific models must now start incorporating neurobiological as well as ecological perspectives into future research so that we can better truly understand what's occurring within all communities relative to this variability and this changing alteration as we move forward in the future for sex differentiation. And the reason this is so important is that it has large implications for human health, conservation efforts, and policy as a whole. Intersex individuals have biological traits that do not fit typical male or female classifications. I apologize. Neuronally, hormonally incongruent individuals who've been previously labeled as transgender may very likely just have underlying intersex conditions. And there's growing evidence and thought scientifically that these two communities, the incongruent and the intersex community, are likely one and the same. Case studies illustrate the complexity of sex differentiation and its medical implications. We've kind of mentioned so far one of these intersex conditions known as complete or partial androgen insensitivity. This occurs in 46XY individuals who develop female typical external anatomy due to non-functional androgen receptors. And this is despite having XY chromosomes and testes, and results in the body being unable to process testosterone. Some individuals who have partial AIS present with a mixed sex traits, both externally and internally. And this further complicates presentation, classification, as well as showing that sex characteristic development, once again, just is not fixed and binary, or else these outcomes would just not be occurring. Another great example is congenital adrenal hyperplasia, known as CAH. These are 46XX individuals who produce excess androgens, and this leads to masculinized genitalia and more skewed masculinized brain structure development. CHH is one of the most common causes of sex atypical development in XX individuals. Early medical interventions, sadly, for this condition and other intersex conditions have often relied on hormonal therapy as well as early surgical considerations. This was meant to be helpful, yet a large number of intersex individuals through longitudinal studies have been identifying that these outcomes being made too soon can lead them to being incongruent with how they truly identify internally, and how a lot of that sexually dimorphic brain development may have occurred during early developmental stages, which produces a real challenge in determining how to best intervene and how do we best assist this community as well as all communities as a whole when it comes to appropriate and equitable healthcare access. You can see to the right a diagram that shows XX chromosome inheritance as well as XY chromosome inheritance, and reasons within those chromosomes that can contribute to female or male development despite being binary. We also can see there is also another condition known as 5-alpha reductase deficiency, and this leads to an inability for testosterone conversion and results in delayed masculinization. 46XY individuals with this condition cannot convert testosterone into dihydrotestosterone, and this is essential for male genitalia development. They may be raised as female due to not having any form of penile gonadal development early in life, and yet during puberty, when that second surge of testosterone occurs, they spontaneously develop penile-based gonadal sex characteristics. And this is more common in some populations across the world than others, such as in the Dominican Republic, where these individuals are known as guevadoses. There's also Klinefelter syndrome and Turner syndrome that help give examples of non-binary sex chromosome inheritance that also contribute to intersex characteristic development. Klinefelter syndrome, 47XXY, individuals may develop testes but have reduced testosterone levels, which leads to partially feminized traits. The interesting thing about Klinefelter syndrome is the very large range in which it actually presents. Klinefelter syndrome has even been documented as just looking like cisgendered male infertility and goes completely undiagnosed throughout that individual's entire life because they end up with penile based development. Their sex organs seem to be functional, and they don't learn until later in life that they're infertile. And then it's just thought, oh, I'm just infertile. Um, and yet they have an extra X chromosome because they've never been karyotyped or genotyped. And there's no way to know that from the outside. It is thought that Klinefelter syndrome has an undiagnosed rate of 44% currently, according to most assessment and statistical models. Turner syndrome, 45XO, where O indicates a lack of a second sex chromosome. Individuals end up having ovarian dysfunction and atypical development. These conditions highlight the diversity in sex differentiation across humans and across other animal species, since this occurs outside of humans as well. The Venn diagram to the right is used to help kind of indicate some of these differences in sex chromosome inheritances that do occur within the intersex community. There are currently 30 classified types of intersex presentations and genetic outcomes, and it's thought that there could be a few more. So ultimately, we want to use this to reframe the trans narrative through more empirical biological evidence and use this to establish a more safe, concrete definition for the trans community known as neuronally hormonally incongruent, to allow ourselves to still keep pursuing forward with research, understanding, and advocacy in a time where language use surrounding trans, gender, things of that nature are being pulled out of scientific research mediums currently. Some individuals that have been historically labeled as transgender may simply have underdiagnosed sex variations, if not the fact as well that these two communities may just honestly be the same. And this is where we need so much more research. Neuroendocrine research suggests that brain sex differentiation may not always align with chromosomal sex. The neuronally incongruence in hormonal function in the brain gives rise to this clearer definition of neuronal hormonally incongruent. And this occurs from that mosaic outcome of masculinized and feminized portions of the brain that we've discussed. Future research publications can use this terminology to continue to investigate links between this community and the intersex community. Examples of neuronally hormonally incongruent individuals further challenge the outdated binary classification of sex. I've mentioned throughout this seminar this mosaic structure to the human brain that occurs through masculinization and feminization of particular cell types and brain regions. And this occurs in all individuals. This is not an intersex or neuronally hormonally incongruent outcome. This functions across the board. And this results in colonization of undifferentiated cells, which is just a fancy way of saying cells that don't have a specific function yet, follow a deterministic pathway of multiple inputs, and then they end up with a range of presentation at the end. As you can see to the figure to the right at the very bottom, when you look at the end of this pathway, you see color differences in these 4 hypothetical outcomes of these cells, which shows that there's a hypo and hypersensitivity to the feminization and masculinization and level to which these influences are occurring within the mosaicism, further indicating just how fully intricate, personal, and unique these outcomes are in all human beings. Hormonal incongruency is defined as an individual's morphological sex and neurological hormonal processing being incongruent with one another and leads to clinically significant distress or functional impairment. Genetic studies have found 21 rare genetic variants throughout 19 different genes so far that have been found to be significantly associated with previous findings of estrogen receptor pathways of sexually dimorphic brain development. What all of this means is that there is direct genetic evidence to suggest that this mosaic formation of the brain is absolutely being altered and impacted within the incongruent community. Genetic variations that have been found associated with both sexually dimorphic brain development and hormonal incongruency include COMT, RYR3, SRD5A2, STS, and SALT2A1, as well as others. These findings hold relevance with the known localized interactions of androgens in the central nervous system. And the reason this is so important to the feminization of certain cell types and brain regions and the range at which that occurs, as we've seen, is because androgens and testosterone, which are one and the same, go on to activate aromatase, which goes on to stimulate estrogen and estrogen receptor activity. MRI studies have found that hormonally incongruent individuals maintain similar neurological response patterns to their binary congruent counterparts. So this is to say, trans individuals and their cisgendered counterparts do not have the same neurological response patterns, whereas trans individuals looked at in comparison to their cisgendered counterpart do have similar response patterns throughout the brain. I've indicated that there are key differences in brain regions from these mosaicisms within the incongruent community that have been identified. And these have been identified not through just MRI studies to show that there's response differences in these regions, but size different studies have shown that nuclei and the overall portion of these brain regions have actual size differences between the incongruent community compared to their congruent counterpart. These occur in the ventromedial nucleus, the medial preoptic area, the anteroventrial ventricular nucleus, that one's a mouthful, the arcuate nucleus, and the bed nucleus of the stria terminus. People be really good at naming brain things, I tell you. Biological sex is just not binary, and medical policies must reflect this reality. Hormonally incongruent individuals require individualized healthcare approaches, and this is not unique to the incongruent or the intersex communities either. The growing field in genetics, known as precision medicine, has been pushing forward for over a decade now and identifying that literally all communities need precision medicine, that there is no true one treatment for one symptom outcome for a large community as a whole. Genetic variation influences the way everyone processes everything. So everyone with anxiety is going to process that anxiety and an anxiety medicine in completely different ways. So this need for precision medicine is not unique to the incongruent or intersex community alone. Research should also be used to inform policies to ensure equitable healthcare access. This should not work the other way around. Policies should never be used to inform research. That's just not how it works. Biology establishes this solid foundation that signaling pathways, development, mechanical function, all of this has a direct way in which it progresses. As a nice example of this, to step outside of all this big terminology and complex systems, think of it this way. I am standing in front of a system that allows me to push a button, it'll move a latch, and then open a window. When I push the button, this stimulates a signaling pathway to the latch, which activates different components of the latch, causing the latch to release, which activates different components and signals of the window, which cause the window to move. This only occurs by pushing the button. You can't tell the window to push the button or open itself and then push the button. And that's not how it works. And this is why research always needs to inform policy. Policy is the window and research is the button. Endocrine variability affects medication dosing, as we've been talking about, as well as surgical outcomes and disease risk. And this is not something that applies to just hormone therapy within the incongruent or intersex communities, but applies to human beings as a whole, as we've indicated. This need for precision medicine and how individual bodies respond to different treatments and different conditions in very different ways very much needs better understanding. Neurobiological differences influence psychiatric care and hormone therapy effectiveness in and of themselves as well. We know that autism, ADHD, and other psychiatric outcomes present differently in congruent male, congruent female, and incongruent individuals across the board, further indicating that we need to better understand what's taking place at a cellular level to better understand the healthcare approaches that are needed to treat everyone and to allow true equitable healthcare to be achieved for all humans. These standardized binary based medical protocols can lead to misdiagnosis and inadequate treatment of individuals. And this causes healthcare providers to do harm that they are not seeking to do. This causes healthcare providers to break a Hippocratic oath that they have no intention of breaking. So we need to not just work to better these communities that need this better healthcare access, but we could also work to help our provisions and providers help create better healthcare opportunities for us as a whole as well, because a lot of them do want to. Increased mental health risks have been highly documented by organizations like the Trevor Project. And this also isn't unique to the incongruent community. Diabetic communities, all other communities, when faced with access issues to gaining treatments, to gaining the medicines they need, end up facing these exact same exacerbations. This contributes to higher rates of depression, anxiety, and suicide in individuals that are denied care. These have physical health consequences. These disruptions in hormonal balances as well can lead to cardiovascular issues, osteoporosis, and even metabolic disorders later in life. This longitudinal long-term health outcome creates a bigger impact to healthcare costs and has high implications from delaying necessary interventions that increase long-term medical expenses, not just for individuals, families, and communities, but for the healthcare system and the state as a whole. There is only a benefit from the small to the large in actively stepping in front of this and providing appropriate access opportunities and care to all individuals. We need to recognize that intersex and hormonally incongruent individuals exist across all populations, and this necessitates the need for inclusive legal definitions. Scientific evidence contradicts the rigid sex classification of laws, even though it sadly took part in establishing some of those. But this is what separates science and keeps it empirical. As science grows, we gain more evidence. We learn how to test things better. We learn how to analyze data better. And we go, hey, these are the new findings. Turns out this is actually working this way. And the field grows, the understanding grows, and legal and policy definitions need to keep up with that growth. Current medical policies often fail to account for sex variation, and this leads to healthcare disparities, and above all, healthcare disparities that are already impacting vulnerable communities that are experiencing varying ranges of healthcare disparities as is. This is important because science can drive systemic change. Research-backed advocacy is key to policy reform. Future medical guidelines should integrate biomarker-driven diagnostics. Legislators, scientists, and medical professionals must collaborate together to ensure inclusive and effective healthcare policies. For future directions of where sex differentiation research needs to go, biomarker-driven studies need to help investigate sex differentiation through genetic, hormonal, and neurobiological, I apologize, got tongue-tied there, markers to better understand the rates at which they're occurring in communities and the impact they're having. Longitudinal research needs to be conducted to better assess the long-term health outcomes of hormonal interventions in hormonally incongruent individuals. We finally need more extensive neuroimaging and epigenetic studies to better understand brain plasticity in response to hormonal and environmental influences. This gives us this idea that we need to begin bridging all of these multiple disciplines together to create a more comprehensive approach to move forward, not just with healthcare for the incongruent and the intersex communities, but for all communities as a whole. And this is going to require collaboration between biologists, endocrinologists, neuroscientists, and policy experts for crucially integrate research into medical practices. Partnerships with public health organizations and advocacy groups are needed to promote inclusive healthcare policies and ensure that these continue to grow in time as further scientific research and evidence continues to better our understanding of these communities. The role of government and funding bodies as well needs to support progressive research initiatives and protect the ability for our healthcare system to better advance itself as science advances and better provide more precise healthcare approaches to individuals across the board. So moving forward, these are the key takeaways of what we've looked at and learned in this seminar today. We've looked at in extensive detail how sex differentiation is a multifactorial plastic process, and it is not a fixed binary within humans or the animal kingdom as a whole. We've looked at how medical and policy frameworks must acknowledge sex diversity also. And we've talked about how future research must integrate biomarker-driven studies as well as longitudinal studies to better assess healthcare approaches. So I'd like to leave all of you, the audience, with a call to action. I always reach out and encourage scientific inquiry, public reform, and equitable healthcare access. Do this by taking information you've learned here in this seminar and the recording that will go out with the seminar once we conclude to Have conversations with family members, friends, community members. Use this to contact legislators, to reach out to state officials and pass this understanding on so that we as a state and a species can keep moving forward and better understanding how to equitably provide care for one another. With that, I'd like to open up the floor for any questions.
I don't have any questions, but I think this was amazing. I had my comments in here. Say it louder for the people in the back.
Thank you. Yes, I was just about to check the chat. Oh, you could hear Twix drinking water? Yes, she was
Yes,
thirsty.
she was
She
very
just
thirsty.
ate a lot of peanut butter. And I would like to, if there are no questions, I would like to kind of wrap up with thanking everyone so much for being here. Attendance to these types of community learning events is essential. Spreading appropriate information is so critical in fighting misinformation and distrust, even between communities and the government. Whereas what we need is a more synergistic relationship between these pieces. So thank you all for being here. Thank you all for caring. And to Jill Jones, who, yes, I was just about to say hello to you. I was going to say thank you so much for popping in. I really, really appreciate you being here. I'm so sorry that, we kind of caught you at the end. There's definitely going to be a recording that'll go out with this, and it'll go on our YouTube, so you can definitely go back and watch this.
And I apologize for being late. My sisters came to visit and stayed longer than we anticipated here. So, is it okay to share the recording with others? We have a pretty active PFLAG group here in Wisconsin in our little community, and so we're wondering if it would be okay to share.
I
Yes.
would adore that.
Okay.
Yes.
We actually have a whole blog page that has extra information where the video will be embedded. So once I email the recording out, I'll send you the link to that so you can share it with whoever you want.
And
Perfect. Thank you.
as an additional follow-up, Jill, on our website, we also, to make bridging this exact topic, especially into community effort to contact legislators, we have email scripts, phone call templates, and a scientific claim refutation one pager on our website that your individuals can use to reach out to their legislators and say, this is the actual science, this is the actual reality, and can have that on hand to help fuel their efforts.
Okay, thank you.
Absolutely. I'm so glad to have you here, Jill. Thank you. Well, does anyone have anything else, or should we all skedaddle for a little Sunday lunch? Oh, and Jill, I'm so happy that even though you were late, it was for a good reason. Seeing family can be so nice. But yes, shall we skedaddle for lunch? I'm ADD.
Let's skedaddle away. Thank you so much, everyone, for joining on your Sunday morning. And Rebecca, thank you so much for just your wisdom and sharing all this information. It's so helpful.
Thank you, Alyssa. That was really kind.